The question is, make a chart with diagrammatic representation showing a restriction enzyme, the substrate DNA on which it acts, the site at which it cuts DNA, and the product it produces. So in this question, we have to answer by making a diagram which is going to represent a restriction enzyme and the DNA on which this particular restriction enzyme acts, and at which site it acts on that DNA or cuts the DNA, and the product that we will obtain once the DNA is cut. So now first let us choose a restriction enzyme. So here the restriction enzyme that we've taken is BAM-H1. You can take any restriction enzyme. Now, the point that you have to keep in mind here is that each restriction enzyme has a particular recognition site. And for this particular restriction enzyme, the recognition site is this GGA-TCC. GGA-TCC. And these recognition sites are nothing but palindromic sequences that are present in DNA segments. Now, let us see this particular DNA segment. Now, in this DNA segment, the BAM-H1 restriction enzyme cuts the DNA at the site between the two guanines on either of the strands. Also, once this cut is made, the product that is produced here is shown below. Now, as you can see, this one strand will be divided into two strands. And now this strand that is obtained as the product has sticky ends. What is meant by sticky ends here? Now, sticky ends are prone to ligation with other DNA segments, okay, or other sticky ends, which will help in recombinant DNA technology techniques. And once this product is obtained, you can see that these sticky ends that are present here will be the ones that are used. So you can see that once the DNA is cut, and the DNA is basically cut from in the middle of the strand. So which means that BAM-H1 is a restriction endonuclease because it cuts the DNA from within the DNA segment. So we have fulfilled all the requirements that are asked in the question. We have mentioned the enzyme. We have then mentioned the DNA segment that it is going to act on. And we have also mentioned the site where this restriction enzyme will act. And at last, we have also drawn the products that will be obtained.